So, hello everyone. I hope the slide deck is visible again, and I welcome you to my talk today about introduction to contrastive self-supervised learning and its application in computational biology and drug discovery. Like the introduction part will be a little bit more than half of the talk, and then we have like the rest for the biofocus. And kind of the idea is for the first part that you kind of get. We will have like a low-level intro, cover all the important concepts, and then look how they are applied. And feel free to ask questions in the middle, or if something is unclear, and I can then we can discuss it. And I guess let's start. Kind of the outline for today is the introduction. We already started with that. First, we will look into contrastive self-supervised learning. Then we will also look in contrastive supervised learning, because this is usually not so well known, but I guess also very interesting. Then we will look into contrastive learning with different modalities. And then finally, we talk about the bio stuff. And then there's an outlook, and let's see then how much time we have left. But the idea is also to have like a discussion, or if somebody has an interesting paper that fits, we can also have a look at it. And so kind of what's self-supervised and how does it differ from supervised? Just a very quick intro. Supervised learning is kind of the traditional setup where we have like data with an associated label. Self-supervised is kind of the same thing, but without labels. And this is kind of a nice figure where you kind of see it illustrated. Supervised learning here in this example, we have kind of two labels represented by the two different colors. And with unsupervised learning, we don't have that usually. And we train a model which kind of captures the structure of the data. So it's then useful in a downstream setup where we usually then have a supervised learning setup again. And this is also something we will see quite often then with the presented contrastive learning setups, that usually we started with unsupervised learning. We want to have a very a well-trained or pre-trained model that already knows kind of or learned the structure of our data, and then we switch to a supervised task where we usually do a fine-tuning step. And this is like one or two slides. This slide and the next one I took from Jan LeCun, and I guess they're very good intro slide. And he has here kind of the comparison to a nice cake, where he kind of asks a question like, how much information do we need for learning? And if you compare it with, or if you take pure reinforcement learning, where you have like very, very sparse learning signals, you have like, it's kind of the cherry on the top. You have just a few bits. Supervised learning, he compares to the icing of the cake. It's a little bit more, but also not a lot. And self-supervised learning can be compared to kind of the biscuit and the cream inside, which is kind of the kind of the big part, but it's a little bit trickier to access maybe in an easy way. But this is what we will focus on today. And this is kind of also the second slide from Jan LeCun. And what you see here is with the traditional self-supervised learning setup, you predict something, you mascot something, and then you try to predict it. So in the end, you feed kind of the pinkish stuff to the model and let the model predict the bluish stuff. And you can do that in all kinds of variations. As you see here, predict the future from the past and do all kind of different setups. And in the end, it comes down to pretend there's a part of the input you know, and the other you want to predict. And this is like just one example. And in the end, there are a lot of different approaches. And contrastive learning is a little bit different. And this is what we will kind of discuss on the next slide. And I guess this is one of the, if you ask me what are the top 3 slides you kind of should take away, because they're very important. I guess this is one of the top 3 slides. And in the end, what we just discussed on the previous slide is we mask something out and we predict something. This is kind of a generative or predictive task, and the loss is measured in the output space. So we have we feed in data and we predict data, and the important stuff is really that this is in the data output space. For example, we feed in here some image, we predict or we feed in here, for example, a grayscale image and predict the colors, which would be colorization, or some kind of autoencoder task, or yeah, this is a mask image or mask sequence, and we predict the output. So this is kind of all kind of the recent advances in NLP or language modeling is in this bucket here. It's generative, predictive, pre-training task, and it works in the output space. Whereas when we do contrastive learning, this is quite different, because we don't measure the loss in the output space, but we measure it in representation space. So we have two samples, and we try to kind of, or we have a loss setup that kind of encodes a relationship, and we want to have that or predict that, and we want to have that represented in the embedding. This is maybe now a little bit kind of hard to grasp, but you will see later with the contrastive examples when we start that this is, yeah, actually a quite elegant way to get a very powerful embedding. Yeah. So let's start with the contrastive self-supervised learning, and maybe one thing about kind of the storyline of the talk. This is how I learned about it. So you will see at the beginning, it's very computer vision focused, because this is where kind of the first papers came out. And the first one is even from Jeffrey Hinton. And then we will look also into kind of sequence data stuff, and then move on from there. So maybe, yeah, I guess it's also very maybe an easier way, because I guess with images it's a little bit more obvious what we do with the data augmentations and stuff like that. But yeah, let's see. I hope so far it's easy to follow. I hope. At least no questions, as I see now in the chat. Yeah, but let's start with the contrastive self-supervised learning. And I have said before that, like, yeah, we don't have labels, but in order for this contrastive self-supervised learning, what we do is kind of we create our labels on the fly, so to say. And here's a very like illustrative example. We have this dog image. We then create views. So this is like a very we call it always a view, and this view is kind of our relationship. We know that those two augmented views are derived from this one image, so they're kind of derived from. Yes. Was there a question? Okay. Yeah, if not, also feel free to post into the chat. Yeah. Otherwise, I will continue. But yeah, we have this kind of parent image. We derive two views, and yeah, let's go on and see what we do with those. So we have then our kind of feature extractor. In this case, it's a convolutional neural network, and those are shared weights here. And what you always have, or what turned out to be very useful, is that you put a multi-layer perceptron at the end, and then use this output embedding there. And what we do is then we have another image. We create here other views from the same image. And yeah, like I said, again, this is all the same network. So kind of, yeah, we use the same network and run all the 4 images through that. And then we have that setup. And what we do then, I try to explain them with this image. So we have then a query. And the query would be, for example, the first dog image here, the gray one. And then we have a positive example, which would be the other dog image. And then we have a negative example, which would be our chair here. And this is in the end our learning setup. So what we do is we say, this is similar to that. So the loss pushes those two embeddings together. And the chair embeddings get pushed away. So we want in the end that those two images are together, the representations of these two images are together, and the two of the chairs are together, but the dog and the chairs should be not together in the embedding space. And this is kind of the learning setup behind it. And the loss we use in order to get kind of push similar things together and push everything apart is the information noise contrastive estimation loss. In the end, it's a cross entropy loss. And kind of on a high level, what we want is what I already mentioned. We want the positive examples, which are here, like the green underlined part. We want the score here to be very high. And when we compare our kind of dog image, for example, to the negative example, which would be the chair, we want the score to be very low. And this is accomplished with this cross entropy loss. And like the green thing is similar images pushed together, and dissimilar are pushed away. So this is kind of the greenish part here. And usually the scoring function here is the dot product. And usually this doesn't have to be always the case, but like in 90%, those embeddings are normalized. So they have unit length of 1. So this is like the dot product ranges from +1 to -1, depending how the two vectors are oriented in space. And yeah, that's actually the whole magic. So we push similar things together and try to push everything away. And this we do like separately. So if we have one image, we want the derived views to be together in embedding space and pushed away from all other views. And this is then applied to every kind of image and the derived views. So far, so good, or are there questions? I hope all good. So then, yeah, then we can continue. Very good, I got it good. So then we can move on. Let me close that. Yeah, and this is the Hinton paper I mentioned that kind of triggered a wave of contrastive learning in the image domain. And what they did is, this is another figure from their paper, but in the end, this is what you already have seen. The X here is our kind of source image. This big T sampled from small t is our transformation. So we have these two transformations, the two views, then we run it through the convolutional neural network and our multi-layer perceptron head, and then we apply the loss. And here they also have that's from the Google blog, they also have this nice animation. And in the end, what I like here a lot is that you see this with the green arrows, what we pull together, and with the red arrows, what we kind of repel. Yeah, that's a very good question, actually. And I guess the distinction is, so the question is, where do CMEs networks and triplet learning sit in the hierarchy of self-supervised learning methods? And there are some big similarities. I mean, CMEs networks, I also don't know every paper out there, but the ones I know is usually that you predict the two kind of those views are the same. And for triplet learning, you would only have like a positive and a negative to your query. And with contrastive learning, you usually have much more, a bigger number of negatives. So it's kind of, yeah, I guess there's no hard distinction, but it's like on a continuous scale, so to say. But usually I can really recommend the SimCLR paper. There are, already kind of 3 versions out there, but the version 1 is very, I guess, easy to read. And they did a lot of ablation studies, and they actually compared, to my knowledge, they're also kind of a triplet loss setup, and the contrastive setup came out on top. But I guess this also maybe depends a lot on your data and your application.
Doesn't SimCLR use ridiculously large batch sizes, like tens of thousands, or am I thinking of another one?
No, usually this is like, you use usually large batch sizes, but I guess also this will depend a little bit on your kind of problem setup. I guess for the SimCLR, they don't go to more than 10K, but I could be also wrong on that, but they have also kind of a nice figure in the paper.
I mean, 10K is what I consider, extremely large. It's not something you can do on a,
Yeah,
single.
no. I mean, it depends on the kind of the data size. So if you work on a problem where you can use very small images, or the data is, or how you can represent your data in a very kind of, yeah, low data amount way, then you can work with that. But otherwise, yeah, you're right. You need more GPUs or more GPU RAM. Yeah. Although I guess there's also at one point also the kind of benefit of large batch sizes is leveling off. Although I have to say, I guess there are some papers out there like Clip, I guess, where they use up to 30K, a batch size of 30K, which is quite substantial. Yeah.
And if I could ask one last question, in SimCLR, the positives and negatives are instances of a image. Like, is a view of an image more similar to itself than on any other image, as opposed to so they don't use the things which are more class-based. I mean, I know there's training based around that, but not in the context of images, as I understand it. And the positive and negative is just positive is a view from the same image, negative is a view from any other random image. They don't do look at clusters similar, another image is just something else or something like that.
Yeah, no, it's really just like, as you said, you have one image, you derive the views, and this counts as positive. But you're right, this has the potential, of course, like if there's another similar dog in the data set, this would be considered a negative, and this can be also a problem. Yeah.
Silly question. I mean, do they do a work of doing instance level and then additional pre-training using the class level or supervised? I mean, I'm guessing people do it. I just haven't seen any works doing it explicitly.
I mean, I guess maybe we discussed it when we talk about supervised learning, because this would be a way how you could approach it. But you could then do it like self-supervised, then go supervised, contrastive, and then go like normal supervised, so to say. So I guess you will see later. Yeah, I guess that fits good to the supervised contrastive setup. But in the end, there are a lot of ways to mix and match. Yeah, the setup components. But I will try to address it on the supervised contrastive part. If I don't do it that clearly, just ask again, please. But I guess it's more clear afterwards. Yeah, so SimCLR, yeah, it's a very nice paper, and I can highly recommend it, even though I reread it a couple of times, and I even find small stuff which is, yeah, sometimes very, very, yeah, well thought through, the experimental setup, and yeah, very, very well done. So yeah, kind of the key findings. They found that the data augmentations are super crucial. So you see what they do is they do a lot of data augmentations, and they also found that you need to have like stronger data augmentations or stronger than you would normally use in a supervised setup. As you can see here, this kind of sober filtering kind of, yeah, looks very different from the real one. And yeah, I guess, and this data augmentation is really crucial, because in the order you want to make the embedding invariant to a specific thing. And what they do with this data augmentations, especially with those strong color augmentations, is that you make the network invariant to the color information, and it then needs to pick up the structure or the object or the object composition in order to be able to discriminate that. And this is a very crucial thing. Like, I don't know, if you want to pre-train your self-supervised network, and you do it that way, that you do very strong color augmentations, for example, and then you have a downstream task, where you want to discriminate different colored objects, you very likely will run into a problem. So this is always data augmentation is a key component, but it can also, yeah, you have to think if it makes sense for your downstream setup. And yeah, what they also do, and this is, I guess, also very nicely illustrated, you don't just do a center crop or whatever, or center crop in the end is kind of the standard approach, but you also do you see these A/B boxes, those are the different views. So you do global and local views, and also views next to each other from the same image. And this we will also, yeah, see later on, that this can be even extended and is then even, yeah, more powerful. And yeah, I mentioned before this multi-layer perceptron already a couple of times. And why this is kind of highlighted is also because adding that on top always helps. Although I have seen also setups where it even works without it, but usually kind of the general opinion is that you should always add it, but you can also check it. And they also have some nice figures in the paper about that. And I already mentioned this one, that they normalized the embeddings because this worked then a little bit better. And yeah, we also talked about this one. The larger batch sizes are beneficial. Yeah, let's go to the unit hypersphere. So like all our features are L2 normalized. So they can be kind of, they are laying on a manifold, which is a hypersphere. And yeah, this is kind of symbolized here with this sphere. And some papers, I'm not sure if this is kind of the final take on that, but there is kind of, you find a few papers where they argue that this kind of hypersphere manifold is very beneficial for linear classification, because then you can separate groups that belong together very easily from the rest. And this is kind of one kind of explanation why this normalized embeddings and this info NCA loss is working that good. And this is kind of, yeah, one intuition. And another one, and I guess this is a very nice view on this setup, is the loss you have seen before. You can decompose it into two kind of mode of actions, and one is alignment and the other one is uniformity. And alignment is, in the end, you see here this airplane pictures that are kind of two views belonging to the same image. They should be very close. They should be aligned next to each other. Whereas everything else, we have here the plane again, everything else should be pushed away. And if you do that with all the pairs you have, so you push away the plane from all the others, then you have here the car and you push it away from all the others, you preserve them, a maximal information, and then everything is uniformly spread out. And this is kind of, seems to be a powerful feature that we kind of can set up with this loss function. And yeah, so this was kind of the SimCLR paper. And now there are coming two extensions of that. The first one is MoCo, and I guess that's, in the end, we already discussed negative samples are beneficial. So we usually increase the number of negatives. So what do you do when you usually, you have not infinite GPU RAM, so you can do one thing, or maybe this is here the end-to-end setup. This would be SimCLR. So we have our encoders, we get the embeddings, then we apply our loss. And what they did with MoCo in order to work around the limitations is that they have here kind of a queue where they keep track of the old negatives and compare them on top. And next to that, they use a momentum encoder. So kind of the weights are updated with this momentum setup. And with that, you can increase kind of the effective batch size. Although this works to some degree, but if you make this queue crazy big, it's also not working anymore as good. This is one option you see quite floating around. And the other one is very interesting, because you will see later on this was scaled to very large scales. And here again, we have the SimCLR setup. You will maybe already recognize again. So we have the feature extractor, the two images run through it, and then we compare them. And what they do with this swapping assignments between views setup is, instead of comparing the features together, features directly, we compare them to prototypes and then use the codes with a swap prediction task. This sounds now, a little bit fancy, but let's have a look at this presentation here. And we have it also on the next slide without the animation to better, get what we're doing here. But we have prototypes that are trainable. When we run an image through it, we look up the closest prototype vector. This is what you see here. Then we swap it, and then we predict it. So let's maybe have a look Okay, I removed the slide, but I hope you see it in here, like when this kind of dashed line. So we have that one, and then we swap it so that the original embedding vector gets compared to this closest prototype. And I mentioned before that this additional fuse is helpful, and they push this with the SWAF setup because they use 2 global fuse. That means like very kind of big crops, and additional small fuse, which are smaller fuse. And I guess they use here 2, and here I guess like something around 8, but I guess they have also some ablations on that. And this seems to be very beneficial and helps a lot during training. So if you can create more fuse of your data, you can also improve the performance. And this is, I guess, a very kind of notable example, and it seems to work very well. But this SWAF setup was scaled up to a 2 billion CNN model, and as far as I know, this is, I guess, one of the biggest, if not maybe the biggest CNN network so far. Okay, what do I mean when I say prototype? So prototype in the end is a trainable vector, and this vector also kind of gets updated. And actually, that's a very good question, because this is like always one thing I ask myself, like, so these prototypes also need to, usually you have a lot of those. I guess they use more than 1,000. Depends also a little bit on the setup. But in the end, what you do is then you have your prototypes and you compare them, you dot product them with the embedding. And then, no, now I'm telling it wrong. But those prototypes are also moving and are also getting updated. And what I always ask myself, and this is what I want to come back at, is I haven't seen work yet where they looked into the or looked into how the prototypes look like, or what kind of features they represent if you compare it to the embedding. I hope this was clear. So in the end, the prototype is a trainable vector or a set of vectors, and they're getting compared with the embeddings.
But what's the goal of having a prototype vector? So what the goal does it serve?
I mean, in the end, this is just another way you could set it up. Kind of the goal is, I mean, they found that it's training, a little bit better than the SimCLR setup. So they were able to achieve, better scores on ImageNet. Although I guess it's always a little bit tricky because they have also this extended view pipeline and stuff like that. But this is just another way you could set up your contrastive learning setup. So let me, there's another question. In these networks, is there weight sharing? If so, do you backpropagate along the single set of weights? So when you look here at those network components, they are all, so those are the same. This is the same network, and you run that through, and the gradient is like accumulated then and backpropagated in one go. So it's along the single set of weights. But we will see later on, for the multimodal stuff, you would have separate weights. But a good question. I didn't mark it here, but this is always the same encoder model. So kind of, this is a very interesting paper because they scaled this CNN model to very large scales. They need a lot of data, but they found out this works, quite good, and it also captures a lot of additional semantics and information. And I guess this is, to my knowledge, this is for sure one of the biggest contrastive learning setups published there. And now we kind of leave the computer vision or the image contrastive learning space, and look look into sequence tasks because they are very interesting for biological applications. And yeah, this is like a very kind of easy example. This is why also the simple is in the name. But what in the end they are doing is they run the data through it, and they have an encoder with dropout mask, and the negative instances are the other one, and the positive instance is the same kind of sequence runs through with a different dropout mask, of course, as a positive example. So this is kind of the low-level data augmentation you can do, and this works also to create meaningful embeddings. And you can also do this supervised, but when you have like labels, but we will talk more also later about that. And this is
I just noted the sentence informers package. They have the pre-training stuff that is just so nice, so nice. Not just saying this just because I'm writing a paper using it now.
Yeah,
they have like a bunch of sort of stuff there, like
they I guess the tree, I'm not sure if they use also the sim CSA, but they have, I guess, several approaches there.
Yeah, TSD is my favorite, if not just because they basically scooped me on it, but
okay, yeah, that's bad then. For sequence tasks, this declutter is like the advanced version of this simple contrastive learning. And actually here you see very nicely in the figure how you can create your views. So you see again the encoder, they have the shared weights, then they have a pooling part, and then also they apply the loss. And here you see very interestingly, you have the anchor here in blue and then the positive sample, and you can sample those two things in an overlapping way, in a next to each other way, or that one is kind of included into the other. And you can also play with the length, and this is what they did there, and they had very good results. And so you can really see you can play with that, and tune your embeddings also in a specific way you want. And similar stuff also works for biological sequence. I already mentioned that, and also for graph data, but this we will also see then in the next section, or in two sections, so to say. Because now we like go over the supervised learning setup, maybe a little bit faster to have more time on the bias stuff. And so let's start with that. So this is now our self-supervised contrastive learning, right? We have our anchor image here, the nice dog. The positive example is also the same dog. And then we have here the other data, and we mentioned it before. It can be that there's another dog in the negatives, and then if we want to classify here cats and dogs, we have a problem because maybe there are a lot of other dogs, and then the embedding is messed up for that task. And if we want to do this kind of cats and dogs stuff, or dog, no dog, then we can, and we have the labels, we can actually do something called supervised contrastive. So we can push all those positive dog examples together in embedding space and move them away from the other animals. And for that, we need the labels, but this can be also a very interesting setup, because kind of what you can do with that setup is that you can maybe create better kind of decision boundaries, because those are pushed more apart in embedding space, and so you get maybe a better classifier setup. And they also have a very nice figure here to compare it to kind of all the three setups. So we have the supervised cross entropy. This is kind of the standard. We have like our label. We have the softmax classifier in the end. When we compare that to the self-supervised setup, we have this contrastive pre-training stage, which you see here in this dashed box, where we do the contrastive loss. And then in the next step, we would add the classification head again, and also do a fine-tuning on the labeled data. And you can also do that with the supervised setup. And I mentioned it already here. Here kind of the hope behind that setup is that here you get an embedding space that is maybe more robust or more better separated, so that the second stage is working better than with the normal cross entropy setup. And actually the interesting thing, or maybe one thing here, why do I mention that? I guess for kind of example for biological data, we have usually kind of data with a few labels. I don't know, we know this is a sample from this organism or it's from this compartment. And what you can actually do is you can create a multi-label supervised contrastive setup. And this is something which actually haven't seen yet, but this would be something that could be very helpful for a few applications. So, and the idea is like you can derive those labels. Actually, there's work out there in the medical domain where they did that from Google, and there it was very helpful actually. But in the kind of small molecule or drug discovery space, I haven't seen it. But this is why I bring it here, because I guess it's a very nice tool to have, because sometimes it fits, yeah. And
I have a question. I mean, does this approach really, is it, I mean, from what I've seen, it a few problems, like they show better performance, multi-class problems, blah, 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 but it's like, I mean, I can't remember the term, it's worse than exponential. It's like, instead of, train time being number of samples times batches, it's number of samples times classes, never mind the input. It's like, it's a gargantuan increase in the training time. I mean, I've seen stuff about it having maybe better results, but it seems like crazy increase in the amount of time it takes to train. Like, have you seen, real world cases of it really being worth it?
Yeah, I mean, you mean like contrastive in general, or
the contrastive in general is more for the fun. I mean, about the supervised contrastive, like instead of doing the softmax, I'm now doing the between all of the pairs between each class and learning all of that.
Yeah, I mean, if you do it like if you use the same classes here, I guess you need to do the experiment, and it's maybe not as useful. But this one medical paper I mentioned, they do it on a lot of different classes, and then they get a very reusable backbone. So you kind of you don't do that one time and use it one time for one stage too, but you can reuse it, and of course then it will make more sense.
Yeah, but like that's the case, I'm guessing, with someone with pretty much unlimited computer doing this is going to be the generic everything model. Yeah. I mean, more.
Yeah, no, I mean this. Yeah, I mean it can be quite substantial. Yeah, and I mean I also can think of cases like I once played with also medical data set on Kaggle, and there I tried first the supervised cross entropy and then the supervised contrastive setup. And what you really see is that the embedding space is here much cleaner separated, whereas this is really very small decision borders or boundaries. And I would bet that this will always lead to more separated kind of clusters that will make your system more robust. But if this is maybe not an issue, and this normal cross entropy setup is working, or the supervised cross entropy, yeah, it's always a trade-off. But I totally agree, from the contrastive learning projects I have worked on, you can train them, yeah, almost forever. They will always improve a little bit. You will see that the embedding space is always moving. It's always a little bit tricky to know when to stop. And yeah, it consumes some GPU time for sure, yeah. And yeah, and this will even get worse when you go into these multimodal models, which we will cover in a couple of minutes. And here, just one example, and I put this here because I like it here, because this is like supervised learning again. And this was like one that pushed recently the state of the art. So this is very similar to this one here. They also train longer, but they could also show small improvements on top. But what I like here to highlight is that they actually use stuff from self-supervised learning, namely this multi-crop data augmentation and this additional projector head here, in order to pre-train the network and then use it downstream again, but in a supervised way. And this also helps. And I find it interesting that you also see kind of that it also kind of, yeah, stuff goes the other way around, and not just self-supervised learning is borrowing stuff from supervised. It can also go the other way around, which I find interesting. And yeah, maybe also these tricks are beneficial for other setups where you usually would not expect it to be helpful. So the next one, like final point before we really go into the bio stuff, and yeah, let's start to have more time for the bio stuff. And I mentioned before, like if you take just 3 slides from this presentation, the top 3, I guess this is also among them. And because I guess this is totally underestimated how powerful the approach is here. And in the end, we now always talked about self-supervised learning over one modality. So just over one kind of images on one setup. But actually, we are not limited to that setup. We can do model compression. We can kind of train a student based on a teacher. And the middle one, which is one I find very fascinating, is you can have the kind of the same data, but for example here, how to phrase that. So here you have kind of the image with in the visible spectra, and here you have kind of a depth map or something else. So you can have two views on the data, like really different views, not just data augmentations, and combine them in a joint embedding space. And I guess this is very something that is very powerful, and it's not used too often. But you can really do a lot of things with that. And also this is one thing that there was also, this was recently also picked up a little bit more, that you have several models. And one very interesting thing, I forgot to put the link here, but what people tried there is, for example, because there's kind of some papers found out that CNNs are more biased to texture, and transformers or vision transformers are more biased to shapes, like on a very high level. But what you could actually do, and I guess I've seen that once, that you actually use here a CNN and a transformer and try to kind of contrast them in the embedding space, so they kind of can, or that you get kind of a joint embedding that picks up both. So I guess you have really here a lot of possibilities to mix and match, and even kind of really tune your embedding space to a specific use case. And this has a lot of potential, in my opinion, and you don't see too often that you kind of mix a lot of those possibilities. But let's move on, because I guess this was kind of the Clip setup, and Clip was not the first one, but it was kind of this big release, and opened a lot of interesting possibilities. And in the end, what is Clip on a high level? Clip on a very high level is we have image data and text data. They are paired. So this is kind of a drawback of this setup, because you need kind of matching data. And then you compare that. So instead of doing one view of an image to the same, or to another view of the same base image, we compare here 2 different modalities. And this turned out to be very powerful if you scale up your model and your data size. And this setup then also could be used in or could be easily repurposed for a lot of downstream tasks. And the way how they approach it is that you train your model on a lot of data, on a very diverse set of data, and then you can simply get, for example, you can easily set up a classifier in a way that you create sentences. Here with this example, a photo of a playing car dog, and then you get the embeddings, and then you run your image through the encoder and take the class from the highest scored dot product in this case. So you can really switch out these text encoder embeddings easily, and you have a totally new classifier, and that proved to be very powerful. And then you kind of train your all-purpose model. So kind of what we mentioned before. This is like a huge run. It takes a lot of GPU hours, so it's very expensive. But you have a lot of possibilities for downstream. And Clip really showed here a lot of interesting applications, from, I don't know, doing almost object detections in images and other stuff, to kind of what was recently also a lot shown on Twitter with this generative models, where you can guide with a text prompt an image creation process. So this is a very multi-purpose setup. And maybe just we will glance over it, over the clip loss, because in the end, you will now see we have one loss part for the images, where we contrast the images to the texts, and another part where we contrast the text to the images. And when you remember the formula from the beginning, this is the same thing, but kind of over 2 modalities. And like image to text has one term and the other way around has another term. This is kind of the basic setup. This setup has also, creates kind of a gap in the embedding space, which can be a problem, not necessarily has to be, but you see that there is not a lot of magic here. And I guess when you look at the kind of high-level pseudocode implementation, you actually see that there's not a lot going on. We run the text and images through the models, normalize them, create the dot product, scale it with a temperature parameter. This is like, I should have mentioned it earlier, that usually you scale the normalized vectors and then you run it through the cross entropy loss. So there's actually not a lot going on, but this kind of setup with a lot of data is able to produce very powerful models. And I can highly recommend this Lucid Rain's Xclip library for really looking into the code of several clip setups, because it's really a very educational repository where you see a lot of modifications and tweaks. And there is a question. That's a very good question. What value does the temperature scaling serve? In the SimCLR paper, they kind of argue that this is to avoid gradient saturation. So you always get kind of a signal that the embeddings get pushed together and get pushed apart, or the positives get pushed together and the negatives get pushed apart. But you can also tweak the temperature parameter to kind of get more dense or more widespread embeddings. So it has, so usually you always use it. In the clip setup, it's even trainable. But this trainable parameter has also downsides because it can be very big, and then, but in the end, you can also tweak your embedding space with that. This is also something some papers investigated, but this is also a way you can tune your embedding based on your downstream tasks. But in the end, it's, it works a little bit better, kind of. This is the high level explanation, and what it does is sometimes a little bit, it's connected to a lot of things, but it's, it in the end changes the learning setup a little bit, and also the embedding space behavior. And this is actually something you can tune, like check a couple of parameters and see how your system behaves. And sometimes it can be that you nicely see this kind of transitions from very dense embeddings to widespread embeddings. If you have later a little bit time, I can also show a screenshot about that. So now we're at the end of the multi-modal setup, and this is one thing I wanted to show. If you remember this top 3 slide, the first one with this generative predictive part and the contrastive setup, this is actually one thing that does it both. They have here a contrastive learning between 2 different modalities, and they do on top also this masked image modeling. And there are some kind of new papers out there that goes in that direction, and this is maybe kind of a possible future setup, although I guess it needs to be proved, or it needs to be shown that this is really beneficial. But I kind of like this mix there. And I guess you could also think that you maybe start out with just this masked image or sequence modeling, because you don't need this paired data, and in the second stage do that on top. So I guess you also have a lot of things to try, but I guess this will be maybe shown the next months if this is like a setup that is really beneficial overall. But this is why I just wanted to highlight that here. And now finally we come to the bio part. And the first thing I mentioned before, graph neural networks are very important for representing molecules, molecular substances. And this is, of course, where this was already applied. And here they used SMILES representation to derive a molecule graph. And here you see the same building blocks as before. You have one molecule, we derive 2 augmented views. Then instead of a CNN or a sequence processing network, we have the graph convolutional network. And then we also have our contrastive loss. And you see here for augmentation, they do a little bit more than just what we have seen before. So they are adapted to the graph setup. They do masking, they delete bonds, or they remove subgraphs. So you have another type of augmentations to apply to graphs. And in the end, the rest is very similar, and the pipeline is also the same. So you have this pre-training stage and then this fine-tuning stage, but you see the concept is very similar, even though the data modality is a completely different one. And yeah, this is a very nice paper, even though it's maybe not state of the art anymore. But what they did, and I liked it a lot, is they did multilingual or multimodal. Yeah, in their case, it's multilingual contrastive learning, in the way that they use two different representations of a molecule, the SMILES and the UPAC representation, and trained on that. And kind of the intuition behind it is that one representation captures different kind of features of the underlying molecule, and this could be beneficial for the created embedding space. And I liked it a lot because I guess this is actually something you could do quite often with the data you have, that you just represent it in a different way, and maybe it picks up more or more interesting features. And yeah, this is why I brought it here, and I still liked it a lot. And this is like a similar one in terms of that they use a different representation, but they do that on so the first one is a graph network, and the other modality is a 3D representation. And yeah, this 3D representation can be like one molecule can have several 3D conformations, and they try to bake that into their approach. And then they use also in the second stage just the 2D graph network and use that for downstream applications. And what they modified the loss in a way, and this is what I really like here, that what I mentioned before, that you have one molecule that has two different 3D representations, and you pull those together and push all the others away. And also another molecule can have also several conformers, and you also put that together. And this is, yeah, I guess a very, very elegant setup to kind of get more information into your embedding space. Yeah, then this is kind of contrastive learning of image and structure-based representations in drug discovery. And this is a very interesting one because they kind of link the molecule structure also with a graph network, and images of cells treated with that structure into a multimodal embedding space. They do that with the loss we have seen before in the modified version. And what they were finding there is that they were then able to do a lot of interesting downstream applications with those two encoders. And yeah, one is like linear probing for bioactivity prediction. So you have your labeled or you have a label attached to your molecule, and then you can predict if they are active on a specific cell type or cell experiment here. And the other one, which is even more fascinating, is that you can do search in the embedding space too, that you have kind of a query image, and you want to find a structure in the embedding space that is responsible for the morphological changes that you get here. And this seems to work very, very nicely. And this is what I then also here put on the slide, because you see here that it's working quite nicely. So here they have the query image, and then the box here is then also the hit. So this is the pair. So you find that, but you also find then similar compounds that have a similar morphological effect on the cells. And as you see here on a very high level, on this kind of low-res image on the slide deck, you even see that this has like a lot of empty structure here, and this is almost there on all the top 5 results. And here it's another compound, and you see that they also look very similar on a high level, and also the matching compound is on the second place. So you can do with these embedding spaces, you can not just do classification, but you can also do, you can search in the embedding space, which could be used for something like that you kind of find similar compounds, or so I guess this has a lot of possibilities. this is another similar approach where they kind of do this contrastive learning. So they call it cross-modal graph contrastive learning. They do this contrastive learning setup we have seen before also, but they also do kind of this masked graph modeling, what we mentioned already a couple of times on top, and also some generative graph image matching, although this is not 100% clear to me in the paper. And unfortunately, I also don't have an ablation study for kind of these contributions of those three components, because I would guess like the heavy lifting is done by the normal contrastive setup. But yeah, it would be very interesting to know. And in the end here, they have like a similar, they evaluated similar downstream applications. This retrieval we already had before, they do something similar. But what I found very interesting there is that they were able to predict clinical outcomes for some data sets, which I found very interesting, and then also property prediction. And so you again see that with this paired data, that this morphological or this image data carries a lot of information for the mode of operation of some, or of the effects of some compounds. Yeah. And this I found this week the first time, this paper. This is actually a very interesting one, because what they did is they linked the molecule and the bioassay description or text related to the assay in an embedding space. And this also was even they even could do zero-shot learning on that, meaning that they were able to predict outcomes on unseen molecules and unseen assays. So they were able to predict kind of the biological activity of novel bioassays just based on this text information. And yeah, I find this very fascinating also because I also posted this in the OpenBioML in one thread, that they're like those huge language models that are out there, that there was a paper that looked into kind of the chemical information that they contain, and this is there, and it's also not bad. So actually there is maybe even ways to link those huge models with a lot of kind of knowledge, not just about chemistry, but in general. But maybe you can use those even here as a text encoder and fine tune it and maybe get even better performance. But yeah, I find this very, very interesting. And this is actually also the last one. And we're already over time, so maybe we just skip that. But in the end, they also looked into contrastive learning for chemical retrosynthesis, where they looked into reaction and the input and the output and the templates, and this was also working there. But yeah. But with that, I will go quickly over the outlook, because I mentioned already a big part of the stuff, and then we can also address further questions or interesting applications you want to discuss. But in the end, I guess it's very interesting that it works that well with two modalities. So I guess it would be very interesting to see what happens when you combine more than that, and that should be easily doable. Then, of course, there's always the big question how to derive it. But we have seen before that with also this supervised contrastive setups, that there are maybe ways where you can actually derive labels in a meaningful way and combine that on top. This I already mentioned before. Can we leverage kind of the knowledge present in large language models? And also those kind of new approaches, setups where you combine kind of this predictive generative pre-training with the contrastive pre-training. Maybe this is helpful. And this was what I mentioned before at those at one of those top 3 slides where I was talking about biases or kind of train your model in a way that your embedding space picks up a very specific part of the data that is very interesting for special downstream applications. Because what you have seen before with this multimodal setups is that you try to train a very general purpose model, but I guess in a lot of setups it's maybe not necessary, and maybe you can tease out or can squeeze out more performance, so to say, on a special application. And I guess this could be something we will maybe see in the future. And with that, actually, I just also want to highlight this multi-modal drug discovery project in the OpenBioML Discord. So there's actually work going on there where we try to address the points up there to some degree. It's still kind of in the starting phase, but if you're interested, feel free to join the discussion, or maybe even the project. And with that, actually, I'm at the end of my presentation. So yeah, if there are questions or if somebody wants to discuss something, I'm still here, and we can talk a little bit. But yeah, thank you very much for your attention.